My name is Fran.
And I'm Jack. We both work in the engagement team at the Wellcome Sanger Institute, a non-profit research organization using the science of genomics to understand biology and improve health. If you haven't joined us before, then Genomics Lite is a series that explores the science that happens here at the Sanger, which is all about DNA codes.
Now, this series streams live on YouTube, so welcome if you are joining us on the live stream. And you can find all of our previous Genomics Lite episodes on our YouTube channel. Now, this session is going to answer the question, how many cells do I have? So we'll be introducing two fantastic guests that work on very different types of cells, and they're going to help us go beyond the numbers and dig deep into what makes those cells unique and why that matters for understanding life itself. But first, Jack, what should we know before getting started?
Well, I suppose the first thing is, what is a cell? Now, most people watching this will have come across definitions of cells before. The most common is that cells are the units that our bodies are made up of, so the building blocks of life. We heard last time in the last Genomics Lite episode how we grow from one cell to an adult human with all of our different parts. And of course, those different parts require different building blocks, different cells to build them. So we have muscle cells, skin cells, epithelial cells in our intestines absorbing all those nutrients, and T cells coming from our bone marrow that help defend us against disease, and lots of other different types of cells as well.
So great. So that gets us to what we're actually counting when we ask that question, how many cells do we have? But what numbers are we talking about?
Well, it could be a very short episode, couldn't it? If I give the number now, I'm going to. The number that's often quoted is that the average human is made of around 30 trillion cells. That's a huge number. That's 3 with 13 zeros after it. But of course, everybody's different, so that number will vary. The average female body contains about 28 trillion cells. The average male body, being a little bit bigger on average, clocks in at 36 trillion. There's also at least the same number of bacterial cells in our body. So that's another 30 trillion or so. But we're going to be focusing on the human cells for this episode.
I guess the question is like, where do we humans fit into the grand scheme of things? How do our number of cells compare with other life forms? Are we big? Are we small? Because there's so many other life forms out there.
Yeah, so this is where we start to get something interesting onto this question. We're at the upper end of the scale, I suppose, but we're nowhere near the top. So the smallest number of cells that a living thing can have is one. Single-celled organisms like bacteria or algae. Then we run right up to the largest animal alive, a blue whale. That'd be somewhere, and I tried to find an accurate number for this. Obviously, it's really difficult to find an accurate number, but it's somewhere in the region of 100,000 trillion, so quite a bit more than we have. Then, I guess, bigger than the blue whale, you've got some absolutely enormous plants out there. You've got some massive mushrooms like the honey fungus networks, if people have heard of those. And they're going to have lots and lots more cells, even than a blue whale. What we're seeing there is that the number of cells scales roughly with size. And we're talking roughly here, but small organisms have few cells and big organisms have lots. Where it gets interesting is that the number of cells scales with size, but the number of different types of cells doesn't scale in the same way. So I listed a couple of examples earlier on: skin, muscle, intestine cells. We don't see that these massive organisms, those honey fungus networks, have lots more different types of cells than other organisms. So again, to give us a sense of scale, single-celled organisms, by definition, only have one type of cell because they're only made of one cell. Most plants and mushrooms are starting to move up to perhaps around 50 different types of cells. That's even those really massive ones. And complex mammals like us and like those blue whales have somewhere between 200 and 300 different cell types. And I'll be really interested to hear from our guests some further information on that. We're near the top of different cell types in terms of the league, human beings. But actually, I've seen a few papers that say that the group of animals that include octopuses or octopi, they actually have more different cell types than we do. So I think it's always important for us to puncture any sense that we're uniquely complex biologically.
Well, I think that's a good time to introduce our first guest. Now, Anna Paredes is a researcher in Rosa Vento's lab at the Sanger Institute, and she looks at groups of cells in the lab to understand what happens in pregnancy. So, Anna, welcome to Genomics Lite.
Hello.
Hello.
Hi, Fran. I'm super excited to be here. Thank you very much for having me today.
You're welcome. Now I'm going to fire off a first question for you. So this episode is all about the number of cells in our bodies, and you work with the earliest stages of human life. So how does the number of cells someone has change throughout their lifetime?
Yeah, actually, that's a very good question, which is actually still puzzling to us as a scientific community, because the reality is that still we don't know. As Jack was saying before, very rightfully, we transition from just being just one cell till these super multicellular, highly complex organisms. But actually, how these numbers are changing across the different stages of life are not clear. We don't know if, like with aging, we just decrease our number of cells, or it's a matter that our cells change in function. So we undergo this aging process that we are all aware of. Why we don't know this yet? Many reasons why. I think is one of them is like that we don't have access to actually the type of samples that would allow us to kind of estimate even roughly these numbers across life. And also because, as you were saying before, each individual is very different. So of course, different female and male, we can have like huge difference that should be taken into account before giving estimations. So this is why actually is so cool, because we have a super nice and interesting niche to work on. Yeah.
Yeah, fantastic. Now, your work is part of the Human Cell Atlas. Now, that's
Yeah.
this huge project, isn't it, which is looking to map all of the different types of cells that make up a human body, how they use DNA and how they interact together. But can you tell us a little bit about the particular types of cells and tissues that you work on?
Yeah, sure. So I work in Roservento lab, as you said before in the introduction. So in the lab, we are super fascinated by trying to understand better the female reproductive system. So in particularly, I work with a very cool female reproductive tissue, as is the placenta, which is a tissue that is made by the fetus as soon as it is implanted in the uterus. And it is essential to actually, as we are all thinking, to establish the maternal and fetal circulation in order to keep the fetus and the mother healthy throughout the whole pregnancy. So these placental cells are a super nice example of this crosstalk that you were mentioning before, because are cells from the embryo that actually are coexisting in the maternal uterus. And they are just nicely doing what they are supposed to do. They are thriving in an environment in which they are actually foreigners. So this is a very cool system to study, and that is very little explored. So yeah.
So is the placenta a bit like you're like growing a new organ? So are there lots of different types of cell in the placenta, or is it like one type of cell, or are there different functions that have to go on in there?
Indeed, very much. There are very different, many different flavors of placental cells. And I think like Alicia in one of your previous Genome Lights, she actually pinpointed depicted very nicely one of the models that she nicely introduced the placental cells. So these placental cells are called trophoblast. And we can have trophoblasts that are just making, as we could see, we could imagine, sorry, the the structure of the placenta that is protecting the fetus, but we have other type of trophoblast cells that are the one that are responsible for actually invading and being in contact with the maternal endometrium. And within those, actually, we know that we have several types of different cell types. Ones that are actually talking to the vessels, the maternal vessels. Some others that are just in charge of reaching the maternal the muscle of the endometrium in order to anchor properly the placenta. So it's actually, many different flavors. Very complex, yeah.
And the fact that it sort of just develops as when you're pregnant, and then, at that time, it's so cool, isn't it?
It
Now,
is.
you study placental cells in the lab in quite a high-tech way. So you don't look at single cells under a microscope, but you use organoids. So can you sort of tell us a little bit about what they are, what they look like, and how you use them?
Yeah, so in the lab we do use placenta organoids, which are basically, and very in short, mini placentas growing in a petri dish. I think this is the best and the most illustrative way of putting it, because it's like mini placentas growing in a petri dish, which actually show the different population of the placenta cells that I was just mentioning, ones that are going to be more in contact with the maternal cells, so that are going to be just mostly structural to the fetus. So this is actually super cool, because that allows us to better understand human placenta, because what happens is that we cannot use other models to actually study human placentation. So we can all imagine that if we want to explore which how many cells do we have in a placenta, and we cannot use alternative models, what do we do actually? Because these are human samples, so it is very tricky sometimes to actually have access to the tissue itself. So we figured out a way in which let's just grow them artificially in the lab, so we can better profile them, and we can actually better understand what's going on in order to better understand how these different cell types of the placenta are being generated.
That's so, so cool. Now, have female reproductive cells been well studied, or is this something that perhaps has, not had much attention perhaps in the past?
Yeah, no, I would say it has now, sadly, no. We don't know much about women's health, unfortunately. Historically and traditionally, women's health has not been really well studied and supported financially. But now we are in another era. We are very hopeful for what's coming. We already know that, and we have growing and growing evidences that human physiology highly differs across sexes. We know that. So now we are in Sanger, and specifically in research lab, we are making a huge effort in order to fill the gap of knowledge that we are lacking, unfortunately, in women's health biology, for these cellular and molecular mechanisms that are actually key in order to sustain women's health physiology. So yeah, we're on that.
That is brilliant because, again, many women struggle to get pregnant, don't they? So is this sort of research going to really help understand and maybe
Yeah.
help it become easier for women to come pregnant?
Yeah, and actually, just let me give you a number. 1/3 of pregnancy complications are related to just placental malfunctions. And we know so little about why that is happening molecularly and at the cellular level, at the single cell level. So, for instance, actually, when your placenta is not able to be developed properly, as different conditions such as preeclampsia that we may be aware of, or like maybe kind of a friend of a family member has unfortunately experienced this preeclampsia, fetal growth restriction. That means that the baby cannot grow enough as the medical guidelines have like appointed. other very severe complications, such as placenta accreta, which is less common and less known. But in these cases, the placenta is highly attached to the uterus of the woman, meaning that at the moment of the delivery, this is a high risk delivery because of the risk of severe bleeding. So actually having a healthy placenta, first of all, having a better understanding how the placenta can be formed and all the things that need to happen to go well and all the things that can go wrong can actually help us to prevent and to better treat all these sort of complications. So yeah, 1/3 is like a massive number. Yeah.
That is, yeah.
Yeah, yeah.
And I guess with, because you're using organoids, like, looking at a placenta must be very, very difficult in a pregnant woman because I guess it's dangerous to interfere.
Yeah.
So would you say that how is your approach examining those cells how is it revolutionizing understanding the placenta?
So I would say that first of all, what we are trying to ground the field by better establishing these models in vitro. Can we actually make better in vitro placental models that can allow us to better answer the questions that we have? So we are doing that by doing by applying single cell approaches that allow us to understand which are the unique features of these organoids at the single cell level. So in order to do that, and I am sure that Jimmy later will explain more about this, we do transcriptomics analysis, meaning that we are reading all the genes expressed at this in each single cell that we have in this mini placenta growing. So by that means, we can actually better comprehend which type of placenta we have in our dish and then make changes or experiments to better adjust to make them more realistic to what is actually happening in the human body. Yeah.
Brilliant.
Well, you've queued me up nicely there, Anna, to introduce our next guest. And I was fascinated to picture that image of these tiny organoids, and we spoke about organoids in our last episode too, but these tiny organoids of a placenta in a dish and how we can study them in a way that is much less risky and pretty revolutionary in comparison to trying to study them while they're inside a pregnant woman. Let me take this point to introduce our second guest, who's working with a very different type of cells, but is going to have lots to say in connection to what Anna's mentioned there. So our second guest today is Jimmy Lee, who is a scientist in the Omar Bayraktar lab at the Sanger Institute. He works as part of this human cell atlas project, looking to map all these different cell types across a human being, but specializing this time in data science and in the computer modeling aspects of this science. So I'm gonna ask Jimmy. Firstly, hello and welcome, Jimmy.
Hello, thank you very much for the introduction.
Yeah. We heard Anna there describing her work with placenta. You work with another very complex part of the body, namely the brain. I want to pay tribute to our question for this session about the number of cells. So I wonder, Jimmy, if you can paint us a picture. One of the cells people might have heard of in connection to the brain is the neuron. How many neuron cells does it take to build an average human brain?
Yeah, so first of all, there is like a recent what they call brain census research about how many neurons in the brain. An average human brain, adult human brain, there are around 86 billion neurons. And fun fact is that actually human brain is what is the brain that still continue to develop after birth. So echoing what Anna just said, our number of cells is dynamically changing throughout life. And then we have to grow from less neuron to adult, which makes up to 86 billion neurons.
Wow, these huge numbers are difficult for my 86 billion neurons to comprehend. But of course, neurons aren't the only kind of cell that we'd find in the brain. So could you just describe for us some of the other things that we've got going on inside of our head?
Yeah, so very interesting. Very interestingly, there are a lot of cell type than neurons. What we usually call the neurons is like the fiber connecting inside the brain between different regions of the brain, but also from the brain to different part of the body. And it's like the fiber. But what people might be less study is that the scaffolding cells, what we call glial cell. And in Greek, glial also means glue or sticking together. So throughout life, they're helping to actually build the scaffolding of the brain into different layer, and then enabling these fiber to go up into different layer for their biological meanings. And also, among one of the glial cell that I like very much and have been studying is called microglia, because it is the cell type that is very similar to one of the immune cell that is very well known, a macrophage. Although it has its immune function that has been studied, but also it is helping with pruning the neuron throughout the development, because these neuron cell like fiber cells, they do have branches to different as I mentioned, different region of the brain, and to accurately, precisely making sure that they are reaching out the right region of the brain, they require cells like microglia to help with pruning their branches. Yeah, so it's very fascinating how the human brain actually works.
I I enjoy gardening, and pruning a hedge is one of the things that perhaps I enjoy most in the gardening. I don't know what that tells you about me, but tell us a little bit more about what's happening there, because I think people perhaps at school will have mostly seen cells as these quite fixed things. Mainly they're looking perhaps at cells down a microscope in a leaf, or a diagram of skin cells or cells in our intestines. I can't imagine any of those cells doing pruning. So say a bit more about exactly how that's working in the brain.
So I would say the field has been mostly focusing on neuron, but actually not much is focusing on cell up to date cell type other than neurons. So the exact like molecular mechanism is still ongoing for study. So probably I think there are sort of like physical interaction between the neuron and the microglia during the growth, and then it helps to prune the branch
Well,
or inhibit the microglia. Sorry, inhibit the growth of neurons. Yeah.
I see. Yeah, so a little different than me going around with my shears then. But you've illustrated something fascinating there, which we heard Anna describe for the placenta, that this is an area that perhaps for lots of different reasons hasn't had as much focus as it's now getting through teams like the team that Anna works in. But the brain has been something that people have been studying and excited about for a really, really long time. And yet there's so much that we don't know, about how this pruning works, or all those other different cells that aren't neurons. Why is that? What's difficult about studying the brain and the cells in the brain?
So, according to the recent brain census, and also a lot of big initiatives like the Human Cell Atlas, uh, basically, it's because the diversity of the cell type that we have in the brain. I've just mentioned that we have fiber like neuron and microglia or different glial cells. Yes, these are like broad terms of the brain of these brain cells. But then we shouldn't ignore that different region of the brain is actually the different cortical region that map back to our different biological function, including cognitive or motor function. And we have more and more evidence now to understand that even for fibers, they have different types of fiber in different cortical layer, which is still we know their identity now, thanks to the advance of single cell technology and single nuclear technology. But then, since we're still at the stage of profiling all of these cell types, I believe that in the future we'll be able to learn about their function. But since we're still at the stage of mapping out their identity, this is the difficulty of or the technical challenge of the field. Yeah.
Yeah. So one that is still really developing. I suppose it's such a complex organ or selection of different types of cells in there.
Yeah.
I think it's worthwhile for our audience just unpicking something that you mentioned there, because Anna talked about this word transcriptomics before, which I think I can imagine most people watching this won't have come across before. You mentioned single cell there. So could you just paint us a little more of a picture? Anna alluded to it earlier, that what we're doing is we're watching different genes in different cells to figure out exactly what they're doing. Help us to understand how that works in the brain. What is it that you're watching there, and what are you trying to gain from watching it?
So to profile the brain, what we're actually profiling, as Anna have highlighted, is the RNA. And what we know about is that whether they are turning on and off, so that we can relate these on and off signal to their biological function. And what's fascinating about the advance of the technology is that right now, we not only just take out a tiny sample out from the brain and to sequence it in bulk. Instead of that, we can start to dissociate them into the single unit of the brain, and then to ask whether the same gene is turning on and off in one cell but not in the other cell, in order to understand how all of these cells, so we dissociate them, but then we put them together to understand how they actually collaborating with each other by turning themselves on and off to build eventually the biological function of the brain. Yeah.
So for people who haven't come across this before, some of our audience will have done, but we've got our DNA in our cells, and the mechanisms within our cells will read certain sections of that DNA and convert that into something called RNA. And that's what you're reading. So instead of reading the big long DNA code, you're reading the bits of RNA. It's picking out of that, and we term that a gene being turned on. Yeah. And so what you're describing there, Jimmy, is that if we look at the pattern of different genes being turned on and off, we can say, we're seeing these cells over here seem to be turning these ones on and those ones off in particular, whereas these ones here are doing something slightly different. Yeah. And so we can say they're different genes. Is that a rough description, right?
Yes, that's exactly, yeah.
Great. Now, we heard Anna describe this from in the laboratory. You know, she's doing it with these organoids, these real samples. You're not working with a brain sitting in your laboratory, are you, Jimmy? So describe, what is it that you're experimenting with? What's the material that's giving you your data?
Yeah, so I would say that, in the lab, yes, there is still a brain in the lab. But thanks to our colleague, they try to use the single cell sequencing technology to basically map out what genes are turning on and off. But doing so, in order to have enough amount of data, what they're doing is that they try to profile, for example, few hundred thousands of cells at a time from multiple samples. And what I'm trying to do on what we usually call the dry lab side, because we don't touch any chemicals and we're mostly working on our computer, we're trying to decode and try to find out the meaning, the biological meaning behind why among 100,000 cells, some of the cells who might have the same identity decide to turn off at the time we sample them, and some of them with again the same cell identity decide to turn on in a given time. Yeah, so
Lots of our audience maybe won't have heard that distinction before of the dry lab.
yeah.
Um, what what is it? What is it practically? Are you still in a white coat and sat at a lab bench, or what? Yeah, where are you working?
Yeah, in my PhD, I used to still wear I used to wear the white coat on the wet lab side. What I always said is that then I take off the white coat and then I work in front of the computer. What is mostly different is right now we're trying to build like borrowing some ideas from historically mathematical formula that are able to run on very fast computer cluster now to decode uh, the the RNA signal of these few hundred and thousand cells. And we use these AI models to understand the meaning behind why these genes are so different, and then to basically try to relate them back to or get an answer back to improve the knowledge.
So, so the computer systems that you're working on and that you examine to get out your experimental data, they're allowing you to shift up the number of cells that you're observing if you were working in a laboratory with chemicals in the same way that Anna does. You've mentioned there 100,000. So that means instead of the few thousand that were being observed in the lab, you're able to look at this bigger number.
Yeah.
I think I wanted to just tie that number together between a few thousand currently dry lab situation, wet lab situation, 100,000 in the dry lab, and then upwards to what you mentioned for neurons across the brain. What was it? 86 billion. There's a few noughts between those two numbers at the moment, isn't there? But you mentioned some wonderful technology in there, and just before we bring Anna back in, I just wanted to hesitate on that. You mentioned AI and you mentioned computer clusters. Could you just unpack those a little bit? Talk about those tools as things in your arsenal as a scientist working at the Sanger Institute.
Yeah, so at the Sanger Institute, because of our in-house computer cluster, we're able to have, for instance, not only one computer, we're actually harnessing so many computer power together and be able to compute some of the very, very complicated analysis, which used to, if on your local computer, would be a month of work to just get out one of the result. Whereas in the computer cluster, we're able to compute everything in parallel, which usually will be just a few hours of work to get the same result. So hence, this is the advantage of us to be able to analyze so many cells at once and at scale. For AI model, again, because these are complicated model that some of the audience might know already, they require a large amount of training data set. While we have few 100,000 cells, that is the amount of data we're talking about. The time to do training on a local computer might take years, might take months. And again, because of the advantage of having these computer cluster, we can do it in parallel. And then we can eventually get our model to be trained very quickly and then be able to answer biological question in a more scalable way.
Right. I'm going to bring Anna back in now as well. So both of you work in really, really high-tech areas. Obviously, we've introduced the wet lab. So Anna's our wet lab high-tech specialist, and then you've got Jimmy in the dry lab. So Anna, how obviously it's quite different areas. So how you and Jimmy both work together on the Human Cell Atlas. So how does it come together, and how do you work together, bringing the wet lab and the dry lab into perfect harmony, so to speak?
Yeah, I would say that everything starts with a coffee together because, and I'm saying this because one of the beauties of the Human Cell Atlas, as Jimmy was very nicely describing, is this huge computational power that we can apply to all the access of samples that we have here at Sanger in order to build these massive atlases of all the cells that are actually building our body, right? But there must be like a endless communication between Jimmy and myself in order to understand what we can actually do with the data that I am generating. What can actually Jimmy do with the data that we are generating at the lab? So this is why I was just saying like a coffee, because we both need to be on the same page. Look, this is, I would just go to the lab, I would take the sample. Let's say that I am working in Jimmy's project with beautiful in the beautiful brain, and I would process the sample in order to get this brain to be converted in single cells. So once, well, in this case, nuclei, but once we have generated our single cell or single nuclei mixture, I would do an experiment that we have mentioned before, this single cell RNA-seq in which, or nuclei, in which we're going to measure all these genes that Jimmy was saying that can be on or off at the different cells that we got. And we are very spoiled here at Sanger because we just sent this experiment to a facility that we have here, which is going to be in charge of all the sequencing. And once that sequencing has been generated, myself and Jimmy, we would get a notification saying, look, your data is ready to be analyzed. And now it's Jimmy's turn, if I can see that. So I will pass it to you, Jimmy.
Yeah,
Yeah,
Jimmy.
I I totally agree with the coffee. And also, when we are analyzing the data, what we are mostly looking at are just the number. And as I said, we have to make sense of this number and then to give biological meaning, especially when there is something that we might be missing during sampling. We will always get back to the wet lab and to have a very, very comprehensive discussion between us. What are we missing, or what should be highlighted in the data so that we can be able to apply the correct model to basically answer the fundamental question that our project wanted.
Yeah, and just to build super quickly on that, because there are like some times in which maybe Jimmy looks at the data, it looks like the cells that he's looking at, they look very much stressed, and that happens. So maybe if Jimmy can tell me this, I can go back to my notes and let's say like, look, that day I noticed that the percentage of cells that were alive were, because of any reason, lower than expected. We can somehow avoid making assumptions that may be just technical, but actually dig into the biology. This is why it's like an two-way street communication all the time, and actually it's more fun.
I'm glad we got fun in there because I think that's so important in our jobs, isn't it?
Yeah.
We're going to have people watching who are thinking about careers in science or careers in a particular area of science. And this kind of collaboration between computer AI modeling and the real world and what's really visible in the lab, that dynamic system, Anna, that you've described there of going, we're seeing something in the model here. Anna, can you verify that or can you check that? Really, really interesting, smart, and fun. I want us to look ahead now. The organoids you described, Anna, really new revolutionary technologies for studying things in the lab. Jimmy, our AI models, building out, what kind of scale we can examine. Lots of change happening, I think, in this type of science at the moment. But I'd like you both to look ahead. What's that change going to mean, and what is the new change coming up? Jimmy, let's come to you first for this. What do you see ahead in the future for the Human Cell Atlas and projects like it, looking to studying all the different cells across the human body?
So as we know more about the cell atlas, or as we profile all of the cells in a human body, I think this resource is very helpful to in general, the scientific community, because then people who might be more so what we usually call ourselves is discovery science. So we profile all the cells, and then we try to discover using a data-driven approach to discover what are the hypotheses. But then we can't just work on ourselves. I think this global effort will really help to encourage more researchers from other institutes to collaborate in order to address some very important fundamental question together. Yeah.
That you've mentioned something there that I definitely want us to allude to on this episode is we've talked about some big numbers today. We've talked about lots of different things, but of course there's some big numbers and different organizations involved in this project that you're in. The Sanger is a special place to work, but there's some wonderful other special partners. Could you, Jimmy, just allude to that? This Human Cell Atlas is a collaboration between lots of different places, isn't it?
Yes, exactly. And we're very grateful to actually have this Human Cell Atlas Consortium that was actually started by Sarah Tagman and also Aviv Regev, who are two very brilliant scientists. And they just come up with this idea that let's profile every single cell in our human body. And then this idea just unify scientists across the world. For example, we already have Human Cell Atlas not only in the UK or US, but also Human Cell Atlas Asia, Human Cell Atlas Latin America. And as the community get together, we now understand that we can't just profile a single cell from where we are. We are also really appreciate the diversity of the sample of every cell coming out from different ethnicity, and be able to address some of the diseases that might more prone to, for example, some of the ethnicity. Yeah.
Yeah, thanks, Jimmy. Very positive image of the future there, and in terms of some of the things that might be delivered from this project. Anna, same question really to you. What do you see in the future for this science, whether that's the kind of practicalities of this science or indeed the outputs from it?
I mean, first of all, I'm echoing everything that Jimmy beautifully explained. This massive effort, as is the HCA, I think has been a turning point for better understanding our human body at the cellular level. That has been it is being revolutionary because we haven't finished it yet. I think that one of the next steps will be start generating human cell atlas for specific diseases, because one of the biggest advantages of being working on this human cell atlas is actually having a reference on how healthy, of course, knowing that we do have differences in ethnicities that can be better improved in the future, but having a reference on how a healthy human body is working at the cellular level. So we are working towards building this healthy reference. Once we have done that, the idea would be starting to actually build these big maps for specific disease in many different contexts. And like the Human Cell Atlas is actually encompassing like the whole body. So cells of the heart, cells of the brain, cells of the liver. So if we start actually thinking on how many diseases we can now profile and better understand by comparing these data to those specifics, is actually mind-blowing. So I think that's going to be the next step.
Yeah, I totally agree with Anna. So I can still remember. So when we were at the stage of profiling every healthy cells in a human body, it was around the time of the pandemic. And I can still remember at Sanger, literally every scientist just dropped their current project and try to curate all the single cell atlas from different labs or different places together and to put together public resources that I always tell people we have unprecedentedly speed up the research to understand unknown pathogen and also to understand how these pathogens actually invade our human body and cause all the diseases. So that everyone can really get out of the pandemic in an unprecedented time period. Yeah.
I'm afraid that is all we've got time for, but it has been such a fascinating conversation that we've had. So thank you so much, Anna. Thank you so much, Jimmy, for such a brilliant discussion on how many cells do we have.
And thank you, of course, our wonderful audience, for joining us today. We hope you enjoyed this session. And if you'd like to watch any of our previous videos, including how do cells grow and what is the microbiome, then you can find those on our YouTube channel or our website, and the link is in the episode description. Genomics Lite is back next month on the 10th of December for our last episode of 2025. And this time we'll be answering, what does DNA do in a cell? Subscribe to make sure you don't miss an episode, and we'll see you next time. Goodbye.